Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology master teacher. So today we are going to do sexual reproduction NCERT questions, few of the selected questions, because a lot of students were asking me, ma'am, we are finding it difficult to solve it or we do not understand the question. So I thought, why not decode it with you? So I have taken the questions and we'll just go through the questions and understand, because yesterday we learned the concept. So today we'll just skim scan through the whole thing and see which are the questions that you're finding it difficult or what is the answer behind it and etc. Okay, now the first question says that name the parts of an angiosperm flower in which development of male and female gametophyte takes place. Okay, so whenever such questions come, students, depending on the mark of the question, I would request all of you to write it. I'll tell you why. Because the question asks about the male and the female gametophyte that is helping in sexual reproduction. So when you are writing such an answer, depending on the mark, see, if it is 1 mark question, you only have to write this. You have to write andrecium and gynoecium. Andrecium and gynoecium, nothing else for 1 mark. But if it is a 2 mark, 3 mark question, you have to write andrecium consists of a unit or a whorl of stamens, right? A whorl of stamens, and these stamens are made up of 2 parts called the anther and the filament. And if the marks are more, you can write little more about the anther, that the anther is bilobed. Okay, anther is bilobed, tetrasporangiate, dithecous. Okay, I'm not writing full. Dithecous, and gives rise to the pollen grain. Okay, filament is a stalk that holds the anther. This is what you can write. Now you can also write that andrecium and gynoecium is the essential part of the flower which actually helps in sexual reproduction of the flower. Yes or no? So this question you can write. At the same time, gynoecium also you can write. Gynoecium is made up of a whorl of pistil or carpel, and each carpel is made up of 3 components called the stigma, style, ovary. Stigma, style, and ovary. And this ovary contains ovules that will later form seeds, right? So this is what you are supposed to write. Depending on the marks, you can keep elaborating it. You can write more about anther, you can write more about stigma, you can write more about style, you can explain a lot about ovary. So students, always remember, when you're writing your boards, when you're preparing for your boards, you always look at the marks. You do not look at anything else. You focus only on the marks, because I'll tell you, I'm not telling that you focus on your marks that you get finally. I'm telling, when you're writing or when you're approaching a question, the first thing that you have to know is how many marks is it, how much points am I supposed to write. If half marks, always consider like this, like let's say half marks is for each line. Okay, each line. So you should see, okay, 2 marks, I'm supposed to write 4 lines and maybe I have to elaborate a little bit. Okay, the same thing goes with 5 marks. Okay, half marks for each point of mine, so I will be writing this many points. This way what happens, the teacher also finds it very easy and convenient, and she also understands, or she gets a notion that my student knows everything and the student has written it in a beautiful flow. Even I have corrected papers of students, and I have the thought process that when the student is writing in point format, okay, when the student is writing in bullet points and when student is connecting each of the point, I know that the student knows the concept, and because of which we'll be able to read the paper in a flow. Got it? So the first question says, name the parts of an angiosperm flower in which development of male and female gametophyte takes place. Okay, they are talking about the ovule as well as the pollen grain. So here I would suggest that after you write about stigma and anther, go for a single line. Okay, go for a single line talking about the pollen grains, because that is a gametophyte. Okay, pollen grains and how they will bear the male gamete. At the same time, go for, when you talk about the ovules, talk a little about the embryo sac. Okay, embryo sac. I'm not going to spoon feed you guys the answer, but I'm just going to tell you what is expected out of you and what you have to prepare, because I know all my students know the concept. It's just that they just want to know, okay ma'am, should I answer like this, should I answer like this? So I'm just giving you a technique to answer. So this way what happens, it is not, you know, a spoon fed class, because you're board students, you should know what to do, right? So now here, next question, second question that we are going to differentiate between microsporogenesis and megasporogenesis. Which type of cell division occurs? Which type of cell division occurs during these events? Name the structures formed at the end of these events. Okay, now we have around 3 to 4 questions. Okay, 3 to 4 questions. Students, I have to also check my audio. Just give me 1 minute. I think audio, yeah. Okay. Okay, now see the first question. Okay, we are going to divide parts by part. Differentiate between microsporogenesis and megasporogenesis. Now all my students knows that microsporogenesis, okay, I'm going to give you a little bit, you know, just about the microsporogenesis as well. Okay, microsporogenesis, students, is the process by which the microspore mother cells, or we can also say the sporogenous tissue, okay, the sporogenous tissue. Now those students who doesn't know sporogenous tissue, you have to go watch my lecture. Okay, sporogenous tissue is the tissue that is present in between the microsporangium, which is a special type of tissue that will undergo meiosis. So the second question of this statement also is answered. Which type of cell division do they go through? They go through meiosis, right? They go through meiosis. So sporogenous tissue undergoes meiosis to form what? They form the microspore tetrad. They form the microspore tetrad, and that microspore tetrad will be covered with callus, and that will be removed with the help of tapetum. Okay, that will be removed with the help of tapetum. The callus will remove that, and they will separate this microspore tetrads, and each microspore tetrads is a potential pollen grain. That is, they will form a pollen grain. So this is microsporogenesis. So microsporogenesis is the process by which we form the pollen grain, or we form the male gametophyte. Okay, the male gametophyte which bears the male gamete. You can tell it like this: with the help of meiosis. Okay, so very simple. Once again, let's revise. Microsporogenesis is where the sporogenous tissues present in the center of the microsporangium undergoes meiosis to form the microspore tetrad, out of which all the microspore tetrad, the 4 microspore tetrads, will become a potential pollen grain. In bracket you can write male gametophyte, and then you can write which bears male gamete. Got it? So write like this, write like this. This way the teacher will know that, okay, the student has decoded this well, because many students, let me tell you, 90% of the students does not understand that pollen grain is the male gametophyte. They think that, okay, pollen grain is the male gamete, because from smaller classes we have studied that, okay, pollen grain means it will go and fall in the stigma, pollen tube will form. Yes, it is correct, but who is going and fusing? Who is the one who is going to do the fertilization? It's not the pollen grain, it's not the exine or intine or the content. The generator cell will bear the male gamete that will go and carry. Pollen grain is protecting this male gamete, right? Which we do not know, but like later classes we study about it. Okay, second question is, what is megasporogenesis? Okay, what is megasporogenesis? Megasporogenesis. Megasporogenesis is nothing but where the megaspore mother cell, okay, megaspore mother cell undergoes meiosis again, okay, meiosis to form, very important, megaspore tetrad. First dyad will be formed, students, then tetrad will be formed, but you can directly write tetrad because you do not want to have the confusion that it is it mitosis, is it meiosis. To avoid that confusion, just go like this: megaspore mother cell by the help of meiosis will form megaspore tetrad. Over. Okay, do not break your head thinking that, oh my god, is it mitosis because it's going from dyad to tetrad. No. Okay, in that write specially that only one becomes functional. Okay, only one becomes functional. So megasporogenesis is when the megaspore mother cell undergoes meiosis and form megaspore tetrads, out of which one become functional and form the embryo sac. Okay, form the embryo sac. So during the formation of the embryo sac, they go through mitotic division. Why am I telling this? Because before the embryo sac is formed, they will have a 2-cell stage. Okay, 2-cell stage. I hope all of you remember this. When I taught you, I drew the structure, no, like this. Okay, 2-cell stage. Then they had 4-cell stage, right? They had 4-cell stage. And then they had 8-cell stage. This 8-cell stage led to the formation of your antipodal synergids. Okay, antipodals. 3 of them became antipodals, synergids. 2 of them became synergids. Okay, 1 egg. Okay, 1 egg and 2 polar nuclei. Yes or no? So this you have to write. If you want to write specifically where the cell division happened, go for this question where they say megasporogenesis is when the megaspore mother cell undergoes meiosis, form megaspore tetrad, out of which one become functional, and that one functional megaspore will form the embryo sac with multiple mitotic divisions. Okay, mitotic division. So this way you will be having a lot of clarity. Okay, when the embryo sac is formed, it will go from a 2-cell stage to the 4 to 8 with the mitotic division, and finally form a 8-cell stage that finally becomes the embryo sac, out of which 3 will be antipodal cells, 2 will be synergids, 1 egg and 2 polar nuclei. Okay, this you can write. Now next part of the question. Which type of cell division occurs during these events? Meiosis is the cell division that occurs during these events. Okay, meiosis. I'm focusing on explaining you a lot more than writing a lot, so you can write your own answers. Just try to understand. Now name the structures formed at the end of these 2 events. Obviously at the end of these events and at the end of microsporogenesis, pollen grains are formed. Pollen grains are formed, which is a male gametophyte. Okay, which bears male gamete. Okay, now at the end of megasporogenesis, what is going to happen? At the end of megasporogenesis, we are going to form the embryo sac. Okay, we will form the embryo sac. This embryo sac will be the female gametophyte, which will be the female gametophyte that give rise to the egg. So egg is the female gamete. Okay, egg is the female gamete. Clear or not, all of you? So I hope so far is good. One second. Okay, done. 3 parts of the question also is done. Okay, going to the next question. Arrange the following terms in correct development sequence. Okay, what is the development sequence? Let's see. Pollen grain, sporogenous tissue, microspore tetrads, mother cell, male gametes. Okay? Yes or no? Yes, done. Arrange the following terms. So we have to see according to, this is what students basically, microsporogenesis, right? Microsporogenesis. So for those who are watching the video today, happy Onam to all of you. I know all the Malayalis will be celebrating this day today. So yes, happy Onam to all my dear students who are watching this video from different parts of the place and who are celebrating Onam. Wish you all a happy Onam, prosperous Onam, and wish your family as well. Okay, so yes, for those students who do not know about Onam, Onam is a very beautiful festival celebrated in Kerala, and it is because I'll give you a very gist of the story, because I feel a lot of students really enjoy knowing the story, because most of the time what happens, we do not know about the festivals that we celebrate, right? So Onam is a festival where King Mahabali, okay, was the king who was ruling the state of Kerala, and everyone was prosperous. Okay, everyone was happy, everyone was prosperous, no one had any problems. Okay, all this was happening, and it is believed that God, got jealous about the fact that no one is praying. Okay, no one is praying to the God, because everyone had so much happiness. Now, it's a very simple thing. Like, if I have a problem, then that's when I remember, okay, I need to call God. I'm just telling the perspectives. Okay, it need not be the same. So same way, God's got jealous that no one is praying them. Everyone has forgotten them because of this king, and Lord Vishnu keeps his foot on Mahabali's head and pushes him down, okay, to the hell. We tell, no, pushes him down to the hell. And Vishnu asked him, since you are such a good person and we had to do this with you, what is one wish that you have? He told, I want to come every year to visit my people. Okay, and Vishnu told, okay, fine. I'm telling about Lord Vishnu. Okay, and Vishnu told, okay, fine, that day will be celebrated as Onam. So it is believed that everyone, every place, okay, in Kerala, every house, front of every house, we will put flowers. Okay, a flower carpet, we will put a flower carpet, which we only make the design, we only put the flowers, and looking at that, Mahabali will come inside and visit us and give us the happiness and prosperity. And if we are suffering from starvation or suffering from any difficulty, he will take away the difficulty. So this is the story behind Onam. So I hope all of you really like listening to such things, because I love listening to such things. I like giving the information as well. So yes, let's go, students. See, we have pollen grain, sporogenous tissue. So I'm going to start with sporogenous tissue. Okay, so sporogenous tissue, this sporogenous tissue is going to undergo meiosis. Okay, or students, in this sporogenous tissue you can also write your pollen mother cell. Okay, pollen mother cell. Okay, that will form what? Obviously it will form microspore tetrad. That microspore tetrad will form pollen grains. Okay, pollen grains will have male gametophyte. Okay, so male gamete, not gametophyte, that is the gametophyte. So male gamete. Done. So here let's put 1. Let's see if we did everything. Okay, 1. This goes as 2, 3. No, sorry, 1 done. 3. Okay, 4, and 5. Yeah, perfect. And when I put the numbers, for me it goes ulta, but yeah, otherwise sporogenous tissue, pollen mother cell, microspore tetrads, pollen grains, male gamete. So you will see this pollen mother cell term given in slash near sporogenous tissue or slash of microspore mother cell. Okay, do not get confused. Mother cells, so it is in such a way, this is your microsporangium. Okay, this is your microsporangium. In the center there is sporogenous tissue. Okay, at the same time, if you see the labeling, okay, of a diagram that is given in your NCERT where they show the different layers, you'll see that the central portion, they have labeled it as microspore mother cell. So this is the same thing. Okay, this is the same thing. So question 3 is done. Chalo. Okay, I've left too many blanks. Now, with a neat label diagram, describe the parts of a typical angiosperm ovule. Okay, they expect you to tell which are the parts of your angiosperm ovule. Got it? Angiosperm ovule. So we have to draw the structure and explain it. So I will draw the structure. It is an anatropous ovule. Now students, different types of ovule are there. Okay, anatropous ovule we are telling because the funicle, chalazal and the micropylar is not coming in a straight line. Okay, so when it is not coming in a straight line, we do not call it anatropous. I mean, we do not call them, let's say we call them anatropous. Okay, see, this is not the best diagram possible, but I couldn't add a diagram, so I will just drawing it. We will increase the thickness. Just give me 1 minute. Yes, it's very humid in Bangalore now. Okay, these are the integuments. Yes, 1 second integument. Okay, now inside this we will draw the embryo sac. Okay, we will draw the embryo sac. So here are the antipodal cells. So I'm drawing a very small embryo sac. Okay, these are the polar nuclei and these are the synergids. Okay, synergids and the egg. Okay, now this is your embryo sac. Now we have to label it. That is the important part of this. Okay, diagram you leave, but labeling, diagram you should know. Okay, diagram you should know, but labeling is more important. Okay, labeling is more important as well. I'll tell you what we have to label. Okay, that looks bad. Okay, yes, so when I have to label, see, these are the integuments. Okay, these two are the integuments, so we will label it both ends. Integuments are, why students, they are protective layers. Okay, integuments, they are protect. Nothing is going properly. Okay, integuments. Integuments are for what? For protection. Okay, for integuments are for protection. Now we have outer integument and this is the inner integument, which I'm not labeling separately. Just know that integuments are two, outer and inner. Okay, this will harden and become the seed coat later. Now if I go here you can see a stalk like structure. Okay, a stalk like structure called the funicle. Okay, stalk like structure called the funicle. Okay, now look at this, the portion where the funicle and the ovary is coming in place. Okay, this attachment, this is called as the hilum. This is called as the hilum. Okay, and in between, what is there? This is the embryo sac. Okay, this is the most important. No, why? That's why we are studying ovule, embryo sac. Now inside the embryo sac, what do you see? I think I'll just label this as antipodals. Okay, antipodals, 3 antipodal cells you have. Okay, this one is the polar nuclei that will later form the endosperm. This is the synergids. Okay, plus the egg will form the egg apparatus. Okay, I'll just directly write there because we do not have a lot of place to label, and with this pen this is how much I can draw. Okay, egg apparatus, and this is your embryo sac. This is the embryo sac. So egg is the most important. Okay, the egg, the pollen grain, the male gamete has to come and fuse with the egg. So this is a typical angiosperm ovule. Okay, typical angiosperm ovule, and you have to remember a few things about this ovule. Is that first thing is that they are anatropous. Okay, anatropous. Most students, one more thing. Okay, where the opening is there, no, this portion where the integuments are absent, this is called the chalazal end. Okay, this is the micropylar end. So sorry, this is the micropylar end. Okay, this is the micropylar end. Okay, micropylar end, and this is the chalazal end. Okay, this is the chalazal end. So opposite to the micropylar end, you will find the chalazal end, and always remember that wherever the integument is absent, that will be called as the micropylar end. Okay, wherever the integument is absent, that will be called as the micropylar end. Okay, now here you have to remember that the anatropous ovule, you have to remember, very important. Okay, it has embryo sac. That embryo sac structure, how they are aligned, all of that also you have to remember. Okay, three things you can never forget. That is your antipodals. Okay, antipodal cells, your synergids or your egg apparatus as a whole. Egg apparatus is your synergids plus egg. Okay, egg apparatus, and then your polar nuclei. Now one more thing all of you can draw is in your synergids. Okay, this is your synergids. Imagine I'm drawing an enlarged structure because there we cannot fit all of this together. Okay, let's say this is your synergids. Remember that at the tip of the synergids there is something called as the filiform apparatus. Okay, filiform apparatus. Filiform apparatus also you can write filiform apparatus you can write, because this apparatus is going to help for the pollen tube to come. Okay, it will tell the pollen tube come this way, because this is where you have to come and fuse with the egg. So this realization, or this pathway, is going to be shown by the filiform apparatus. Okay, you don't forget this. And wherever the polar nuclei is there, that whole thing you can call it as the central cell. Okay, this whole thing you can call it as the central cell. So this is what you have to remember in your ovule, very importantly. I hope that was clear. Now next one. What is meant by monosporic development of female gametophyte? It's a direct line of NCERT, and I'll tell you what is each word meaning. Now students, mono means single. Okay, monosporic means a single, like a single spore, something like that. No, so this is actually one spore. Now ma'am, what spore are you talking about? Very simple. Your megaspore mother cell, okay, your megaspore mother cell did meiosis. Yes or no? It did meiosis. After meiosis, it gave rise to, like microspore, it gave rise to megaspore tetrads. Agreed or disagreed? Okay, megaspore tetrads. In that tetrad, okay, in the tetrad that the megaspore mother cell formed, in the tetrad that the mother cell formed, what happened? 3 of them got degenerated, or 3 of them became non-functional. Yes or no? 3 of them became non-functional. Only one megaspore mother cell, only one megaspore, okay, only one megaspore became the embryo sac, or became the mature female gametophyte or gamete, right? So when the development is happening from only one megaspore, okay, from only one spore, that kind of development is called as monosporic development. Okay, so in your textbook you can see that there is a statement given about monosporic development, that when only one becomes functional, only one megaspore becomes functional, we call it as monosporic development, because other 3 of them also gets degenerated, or other 3 of them also get non-functional. Understood? So one megaspore making the entire development is called as monosporic development. So one spore, that is the meaning. Understood? One spore. So megaspore mother cell undergoes meiosis to form megaspore tetrad, out of which 3 becomes non-functional, only 1 becomes functional, to end up forming the embryo sac bearing the female gamete. Okay, students, bearing the female gamete, you have to tell. Bearing the female gamete. Done? Got it? Okay, very easy question. Okay, very easy question. I leave solution place and then I write it there. Okay, with a neat labeled diagram. Okay, with a neat labeled diagram, can you explain 7-celled 8-nucleate nature of female gametophyte? Okay, so we will take all the place. Same thing, students. But here we are going to draw your embryo sac. Okay, they're asking us what? We will write what they're asking us. Otherwise, we'll forget. Right? What are they asking? 7-celled 8-nucleate. Right? 7-cell 8-nucleate is the question. Yes, 7-cell 8-nucleate. They want us to explain. Okay, before I explain, before I tell anything, first I have to draw a diagram to explain. Otherwise, you will not understand, right? You will be like, "Ma'am, what is this ma'am telling?" Like, if you're a student who's only looking at the questions without learning the chapter, or want that last minute, like, "Ma'am, my mid-sem is tomorrow and I just want to go through the whole revision. That's why I do not know the lot of concept, but I just want you to make me understand for marks." So for that, students, I am going to draw and explain, so it will be easy for you to remember for a longer period of time. Okay. You would have understood what I'm drawing, right? It's the embryo sac. Okay, it's the embryo sac. What is missing here? Egg is missing. This is the egg. Okay, so this is our structure. Okay, now students, the question is, can you explain how it is 7-cell age nucleated? Okay, first of all, let's label these parts. These are none other than your antipodals. Antipodals, yes or no? These are your synergids. Synergids. This is your egg. This is the central cell. Okay, and these are your polar nuclei. Polar nuclei. What I missed to draw? Filiform apparatus. In big diagrams you can draw, okay? In small kuti diagrams you cannot see this. Now the question, let's come back to the question. Why is it 7-cell 8 nucleate stage? So simple thing. How many nucleus do they have? Let's just count the nucleus, okay? 1, 2, 3. So 3 antipodals are there. So they will have 3 nuclei, 3 cells. Okay, okay, ma'am. 1, 2. Okay, 1, 2. That is 2 nucleus is there. So here central cell have 2 nucleus, but central cell or polar nuclei is considered as a single cell. That is, they are considered as a unit, like you and your sibling is considered as one unit. Why you are considered as one unit? You both are two different individuals, both of you have different characters, but you will be considered as one unit. Okay, same way polar nuclei, even though 2 nucleus, each of the cell will have a nucleus, we will consider them as one single cell called as the central cell or polar nuclei. Okay, because even when fertilization happen, one male gamete will come and fuse with both of them to form the triple fusion. So they are considered as a single unit. Okay, so this will be considered, they'll have 2 nucleus, students. Okay, 2 nucleus but a single cell. Okay, 2 nucleus but a single cell. Now egg will have 1. Okay, synergids will have 2. So 3, 4, 5, 6, 7, 8. 8 nucleate, no doubt ma'am, 8 nucleus is there. 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus is there. Cell ma'am, 7 cell. 1, 2, 3, 4, 5, 6, 7. So this is why we call our embryo sac as 7-celled 8 nucleate stage. This is how the embryo sac is there. Okay, this is how the angiosperm embryo sac is present. Got it? 7-celled 8 nucleate stage where the central cell will be considered as a single cell. Understood, students? So with the help of a diagram, they have asked us to explain. We have done the explanation that the polar nuclei, okay, polar nuclei will be considered as a single cell. Okay, one single cell but with 2 nucleus. Okay, with 2 nucleus. Clear, all of you? Chalo. Next question. What are chasmogamous flower and can cross-pollination occur in cleistogamous flower? Give reasons of your answer. Okay, I want to show you a chasmogamous flower, but I don't know if I'll be able to do that. Okay. I think all of you would have seen this flower. Okay, these are the chasmogamous flower. Okay, these are the chasmogamous flower. I think all of you can see. Okay, like this. Yeah, can you see? Yes. So these are chasmogamous flower. Now they are asking about 2 things. Okay, they are asking what are chasmogamous flower. Students, so according to your NCERT, under autogamy. Okay, under autogamy, 2 things they have given: chasmogamous and cleistogamous. Okay, cleistogamous. Now the question asks about what is chasmogamous flower. So according to your NCERT, chasmogamous flowers are the flowers which have exposed anther and stigma. Okay, exposed anther and stigma. And these people are going to open. Okay, they are going to open during maturity. Okay, they are going to open during maturity for cross pollination to happen. See, they'll allow cross pollination, but only during maturity they are going to open and help for cross pollination. Okay, now the second question is, what is cleistogamous flower? Now cleistogamous flowers are the flowers that do not open at all. Okay, do not open. Even when they open, they are not facilitating a cross pollination, so they do not open, students. This kind of flowers, there is like very nil chance, okay, negligible chance, or very rare chance for cross pollination to happen. So they will always undergo self pollination. Okay, they will always undergo self pollination because of very simple reason, that anther and stigma is in close proximity. Anther, stigma in close proximity. Yes or no? Close proximity. So very simple, students, that clistogamous flowers are the type of flowers that do not open, and even when they open, they do not favor cross pollination, because you have to remember that the pollen grains have to be carried by agents, right? They cannot walk and come, they're not motile, so they have to be carried by agents. And when they are being carried by the agents, this flower does not facilitate like maximum opening, so that the pollen grains can be dusted. So they will open very slightly, and that will not allow cross pollination. So for clistogamous flower, it is very rare that cross pollination happens. So clistogamous flower will always go through autogamy. Okay, always go through autogamy. Clear, all of you? Yes or no? You want to see clistogamous flower also? Clistogamous flower. There's no good example, student, but I think you can see this. Okay, these are clistogamous flowers. Okay, they do not open. Can you all see how they are? They do not open. I think the brightness is too much. Focus. Yeah, you see, they do not open at all. Okay, they do not open at all. So because of that, they will not be able to do. But chasmogamous flowers are not like that. Chasmogamous flowers do open during maturity, and there are chances, and like compared to clistogamous, there's definitely a lot of chances for cross pollination to happen, and it can happen with the help of your insects mainly. Okay, because they will be colorful. Okay, they are a little showy flowers only. So entomophily can happen. Okay, entomophily can happen, anemophily also can happen, wind also can happen. So what they do is, during maturity, they will expose their stigma and anther. Okay, they have an exposed stigma and anther, and they open during maturity. But clistogamous are not like that. You saw now how they are closed, so they will not allow for cross pollination to happen. Chalo, that is done. Next one. Mention 2 strategies evolved to prevent self-pollination pollination in flowers. So students, continuous self-pollination leads to inbreeding depression. So this is how you have to decode these questions. Okay, inbreeding depression. So the question states you to tell 2 outbreeding devices. Yes or no? The question is directly asking you tell 2 outbreeding devices. So inbreeding depression can be avoided by the plants itself. The plant is like, I know the problem, I will deal with the problem. So what the plants will do, first thing they will do is they will start forming unisexual flowers. Okay, unisexual flowers. Now why did inbreeding depression happens? Because of continuous self-pollination. Okay, continuous self-pollination. Yes or no? Continuous self-pollination. Because of this continued self-pollination or continuous self-pollination, what is going to happen? The flower will go through inbreeding depression, or the plant will go through inbreeding depression, where there is no variation at all. So the plant will decide that no, I want to come out of this depression. I am going to do something to avoid self-pollination from happening. So first thing they will do is they will form, they'll start making unisexual flowers. So this way, what is going to happen? Inbreeding depression will not happen. Second thing that they do is they will change the position. Okay, they will change the position of anther and stigma. Will change the position of anther and stigma. Now change the position means what? First condition, this is your stigma. Okay, first condition, this is your stigma. Got it? stigma style ovary. Okay, stigma style ovary. In the first situation, your anther was like this. This is how normally anther will be, students. It will be a little above from the stigma. Okay, this is how it was. Got it? In the second situation, the plant itself will do one thing. The plant will change the position. Terminologies are there for this, but you do not have to know. Okay, so it will reduce the size. Will increase the size of the stigma. This way what will happen, the anther will not fall on the stigma. The chances of anther falling on the stigma is almost negligible. So they will change the position of anther and stigma. This way what will happen, self pollination will not happen. Okay, now one more step extra I will tell you. Okay, 1 or 2 steps I'll tell you extra. So if you feel that ma'am this is not easy, I want this step, you can choose. Then students, receptivity, that is self incompatibility will start. Self incompatibility. What is self incompatibility? That is when the pollen grains from this stigma, okay, this anther falls on the stigma will start rejecting it. Okay, the stigma will start rejecting it, understanding that it is my own pollen grains, it is falling from my own plant, and they will start rejecting the pollen grains. So self incompatibility happens. Okay, this way what will happen, stigma's receptivity, pollen pistil interaction is there, no, that interaction will not happen. Okay, that pollen pistil interaction will not happen. This way the stigma and the pollen, everyone will be happy that okay, we have avoided self pollination from happening. Okay, then the fourth step is the receptivity. Okay, receptivity. What is receptivity? That is the pollen and the pollen anther. Okay, the anther and the stigma will get matured at the different times. Stigma and anther will get matured at the different times. Now imagine we cannot change the position or such a condition is there. What will happen? Pollen grain will dehiscent earlier, maybe. Okay, this will dehiscent earlier. This will get receptive later. So in this situation also, definitely students, obviously pollen pistil interaction will not happen because stigma will be like, wait a minute, I have not matured. Pollen will be like, wait a minute, anther has already dehiscent and I am already out. Like, you know, the pollen grains have started moving everywhere. So pollen will be like, I got matured, and stigma will be like, wait, I am not mature. So obviously self-pollination cannot happen. These are the 4 steps. These are the 4 outbreeding devices. So you can remember any one of it. That is forming of unisexual flowers, very easy to remember. Changing the position of anther and stigma, decreasing the size of the anther or increasing the size of the stigma. Third one is still self incompatibility, rejecting their own pollen grains. Fourth one is changing the receptivity. The maturity period of anther and stigma will change. This way self pollination can be avoided. Got it? Chalo, next one. What is self incompatibility? What does self pollination not why does self pollination not lead to seed formation in self incompatible species? So just now I told you guys, so this is your stigma. Okay, this is our own stigma. I've taken few selected questions, students. I've not taken all the questions. Okay, I've taken only few selected questions which you might need help. Okay, this is, let's say, orange color is the own pollen. Okay, this is the another pollen. This is another pollen. Okay, this is another pollen. Now self incompatibility means what? The stigma knows that this is my own pollen and I am going to reject it, but these two are from different places, and out of which I find this one as the rightly compatible, so I will allow it to start forming the pollen tube. Understood? So I will, after the chemical dialogue, I will allow for pollen tube germination. Okay, pollen tube germination. Is this clear? Okay, pollen tube germination. So obviously in self-incompatible species, the seed formation will not happen because the pollen grain got rejected and the pollen grain was not allowed to form the pollen tube. Now what, students, where is the ultimate of this pollen tube? It is going to form the pollen grain all the way down and it is going to enter into the egg apparatus, right? It is going to enter through the synergids, which will carry the male gametophyte, maybe in a 2-cell stage or a 3-cell stage. If it is in a single cell stage, it will directly form the embryo, and that embryo the zygote, okay, then the embryo. That embryo will directly form the monocot, dicot, whatever, cotyledons and other things, right? They will give rise to plumule, radicle, all of this we studied last class. Or in the 3-cell stage, 2 male gametes will be there. One will form the endosperm, one will form the embryo. These two will be happy. Even self-incompatible. Pollen grain only got rejected. Then how will pollen tube germination happen? How will the male gamete meet the female gamete? So this will not happen. Understood? This will not happen at all. Next question, very good question. Okay, what is triple fusion? Where and how does it take place? Name the nuclei involved in triple fusion. Okay, so yesterday I had written one box. So triple fusion is nothing but, this is your, okay, I love to teach with diagrams, okay, because diagrams make you understand the concept better. So student, this is your synergids. Okay, this is your synergids. And this is your polar nuclei. Okay, this is your polar nuclei. Now this is the filiform apparatus. Filiform apparatus is very important when you're talking about double fertilization, because this is the fellow who will be like, come inside, have some tea and samosa, all this fellow only will tell. Okay, so students, look here, what is going to happen? The pollen tube germination has happened. No, the pollen tube will enter one of the synergids, okay, with the help of your filiform apparatus. So in this, there is going to be one male gamete, one more male gamete is going to be there. Why two male gametes have come? It's very simple. One vegetative cell plus two generative cell stage has come. This is called as the 3-cell stage. 3-cell stage. So during this 3-cell stage, two generative cell means one male gamete, one more male gamete. One male gamete will go and fuse with the egg. Okay, one male gamete will go and fuse with the egg. Another male gamete, let's draw it in different color only. Okay, another male gamete, purple color, I don't think it will be visible. Okay, green color. Okay, another male gamete will go and fuse this. This is an entire cell, no? This is an entire cell. One second, students. Problem with this is, okay, oh, it went off. Just give me 1 minute. Okay, this is the pollen tube. Okay, no way to go back, no? Yeah, this is the pollen tube. Is ready to enter. I've drawn a big pollen tube. Okay, one male gamete is there, another male gamete we are going to draw. Oh, I'm choosing the same colors again and again, and this is one more male gamete. Okay, this is so sorry. So sorry. Yeah. So one has gone and fused with the egg. Okay, one has gone and fused with the egg. Okay, the one that fused with the egg, that is going to form the embryo. So triple fusion means what? Very simple, students. Double fertilization happens. That is, the actual male gamete, the male gamete that we decided in the 2-cell stage, will come and fuse with the egg and form the embryo. This is normal situation. Okay, but what is going to happen? One more male gamete, that is, after reaching at one point, the generative cell will split again, or the generative cell will go to multiply and form one more male gamete. Now that male gamete will go and fuse with whom? We have only one egg. We have only one female gamete. Where what will happen? That time they will tell that, okay, you know what? This is a central cell. This is a polar nuclei. So what you can do is, this male gamete will go and fuse with the polar nuclei, and this whole thing is called as triple fusion. Okay, because two nucleus is fusing, right? Two nucleus is fusing, so they become triple fusion. No, three people, right? One, two nucleus with one more. So very simple, students. Triple fusion is when the one male gamete, okay, one male gamete fuses with the polar nuclei, forming the pen. So now this will give rise, okay, this will give rise to nothing but pen. Pen is primary endosperm nucleus. Okay, and later the fate of this is going to become the endosperm, and this is going to give the nourishment for the growing embryo. So triple fusion is this, okay, where the male gamete fuses with the polar nuclei to form the pen, that is primary endosperm nucleus, whose fate is going to be endosperm, who is going to give the nourishment for the growing embryo. So this is what is triple fusion. Now where and how does it take place? It takes place in the embryo sac itself. Name the nuclei involved in triple fusion: polar nuclei plus the male gamete. Understood? Understood how it is triple fusion? Pen, because it is going to form a triploid structure, student. The endosperm, the pen is going to be a triploid structure, because understand, three nucleus fusing is happening, and this pen's fate is going to be, it is going to form an endosperm, and this endosperm is going to provide the nourishment for the growing embryo, telling that you take whatever nourishment you want, you grow better. Okay, embryo is going to be formed how? This people are going to give a rise to a baby, that is going to be the zygote or the embryo. Understood triple fusion? Very easy peasy question. Okay, very easy peasy question. This together, because two fertilizations are happening at the same time, we call it double fertilization. So one male gamete, or the first male gamete, you can tell, we tell usually first male gamete, one male gamete. Okay, one male gamete will fuse with one egg, and that will form the zygote. That zygote will later form the embryo. Okay, embryo. Now the second male gamete, okay, the second male gamete, which was seen in the 3-cell stage. Okay, second male gamete will fuse with the polar nuclei present in the central cell to form your pen. That is primary endosperm nucleus, whose fate is going to be endosperm. Okay, endosperm. Now this endosperm will provide all the nourishment for the growing embryo. Understood? All the nourishment for the growing embryo. Nourishment. So since these two fusions are happening at the same time, okay, at the same time, this we will call it as double fertilization. Okay, two marriages are happening at the same time, so we will call it double marriage. No, nothing like that. Double dhamaka we tell, no, double fertilization. Got it? This is your whole triple fusion and all the nuclei that are involved with it. Now, okay, differentiate between hypocortile, epicortile, coleoptile, coleorhiza, integuments testa, perisperm, pericarp. Okay, now very important, hypocortile and epicortile. So students, if I draw, see, the plant is going to have an embryonal axis. Okay, when the seed is going to germinate, it is going to have an embryonal axis, and they are going to have two cotyledons. This all of you know. No. Now what I'm drawing is the embryonal axis. What I'm drawing is the embryonal axis. This embryonal axis that is emerging above the cotyledon. Okay, this is the cotyledon, my hand is the cotyledon. Whatever is emerging above the cotyledon will be called as the epicortile. What is it called? Epicortile. This epicortile will give rise to plumule. Okay, it will give rise to plumule, and the plumule will form the shoot. Whatever is coming above the soil, whatever is coming above the soil. Now whatever is below the cotyledon, okay, whatever is below the cotyledon will be called as hypocortile. That will form, that will terminate. Okay, terminate we should tell, it shouldn't tell the word form. You can tell the word, the epicortile terminates in plumule, the plumule becomes shoot. Same way the hypocortile will terminate. Okay, the hypocortile will terminate as root, and not root students, radical, that will form the root. Understood? So this is the difference between hypocortile and epicortile. Epicortile is the layer above the cotyledon which will terminate as a plumule that will give rise to shoot, and hypocortile is the region below the cotyledon which will terminate as radical that will form the shoot. Okay, that will form the root. Okay, done. This is done. Coleorhiza and coleoptile. This you usually see in the seeds. Okay, in the cotyledons you will be able to notice that there is a portion that is extending that will protect the plumule, and there is a portion that is going to protect the radical. Okay, there is a portion that will protect the radical. So the portion that is going to protect the plumule is called as coleoptile. Okay, students, why is this? This is because the seed has to go through sometimes a very unfavorable condition. So during this time we have to make sure that the seed will germinate. Have you all seen there are plants that grow on concrete? Okay, there are plants that grow on tar roads, there are plants that grow on the rooftop. Why? How is this happening? Because the root cap is very good, at the same time the seed is able to have such protection, like your in monocot. Now if you see, you have your cotyledon, one single cotyledon. Scutellum is there to divide, to do the separation between your embryo and the endosperm, like that. Coleoptile, coleorhiza, all this is there. Root cap is there. This is the protection that the plant is giving, telling that okay, don't get damaged. You give rise to the you give rise to the root and shoot. This way, what is going to happen? They are not going to get damaged at any cost. So, coleoptile is the portion that is going to protect the plumule. Okay, and coleorhiza is going to be present near the radicle. Okay, now difference as such, like there's no definition, but if you want to write it, you can write this. Okay, coleoptile and coleorhiza. Yes, clear, all of you? Coleoptile and coleorhiza. Yes, coleoptile and coleorhiza. Okay, so this is one difference of it, that this is going to be above the soil, which is going to give rise to the plumule or the shoot. This is going to remain in the soil, which is going to give rise to the root. Now, if you want to write a little extra, you can tell that this will protect the shoot, this will protect the root. All this you can write, and you can write where they are, like one is above the soil, one is below the soil. Okay, one is above the soil, one is below the soil. You can write that. Yes, clear? All of you? Okay, fine. Okay. Yes. Then integuments and testa. Students, integuments is a protective layer of the ovule. Yes or no? Integuments. Okay, one second. I think here I didn't leave, so here also no place. Okay, integuments is a protective layer of your ovule. Protective layer of ovule. And testa is the protective layer of your seed. Okay, testa and tegmen is made. No, testa and tegmen is made. Testa is the outer covering, tegmen is the inner covering. This is to protect the seed. So, integumenta is a protective covering or a protective layer of your ovules to protect the ovules, and where the integuments are absent is called as micropyle. Testa is the protective covering of the seed. That's all is the difference. Okay, that's all is the difference. Next is perisperm and pericarp. Students, perisperm is a persistent nucellus. Persistent nucellus, okay, which is seen in beet and black pepper. Beet and black pepper. Pericarp is the hard layer of the fruit. Hard layer, or we can tell fruit wall. Okay, fruit wall. The ovary wall will become fruit wall, so that fruit wall is called as your pericarp. To protect the fruit, no? The fruit wall is called the pericarp. So, perisperm is a persistent nucellus. Nucellus is what? It is present in the embryo sac. So, when I draw the embryo sac, now on the top of the embryo sac, there are specialized cells which is called as nucellus, which provides the nourishment for the growing embryo. Okay, do not get confused with growing embryo sac and growing embryo. This is the embryo. This will later form the, like that is after fertilization, you will see the embryo, endosperm and all of that. Embryo sac. So, nucellus is a cell that will give the nourishment for the growing embryo sac, and those, okay, that will persist and that will stay like that only. It will stay like that without, getting degenerated. So, that persistent nucellus is called as perisperm, which example is beet and black pepper. Then, pericarp is a hard layer or the fruit wall. The ovary wall will get modified to fruit wall. That is called the pericarp. Clear or not, all of you? Okay, clear? Now, why is apple called false fruit? Which part of the flower forms the fruit? Very simple definition, students, for false. This is the last question that we'll be doing. Okay, false fruit. Okay, false fruit and true fruit. How do I differentiate, ma'am? Right, this question you will have, ma'am, how do I differentiate? Students, very simple. True fruit. What is the fate of the ovary? The ovary has to become the fruit. Everything has a fate. Okay, everything has a fate. If they don't follow the fate, then they are not true. Okay, ovary will become fruit, ovule will become seed. This is the fate of them. So, if the ovary becomes the fruit, or in true fruits, the ovary will get modified to form the fruit. This is called as true fruit. Now, what is false fruit, ma'am? Any other, okay, sorry. Any other part other other than ovary is forming the fruit. Okay, is forming the fruit, then it will be called as false fruit. Now ma'am, example, why did they talk about apple? Students, apple, the portion that you consume, no, this portion that you consume, the portion that you consume, this portion is actually the thalamus. What is it? Thalamus. So the thalamus has formed the fruit. So obviously I'll call it false fruit, no? Ovary was supposed to form the fruit. So true fruit is where the ovary becomes the fruit, and false fruit is any other part other than the ovary if it develops into a fruit. Okay, that will be called as false fruit. So the thalamus in the apple has formed itself as a fruit. So that's why we call it false fruit. They are asking which part of the flower. Thalamus. Okay, thalamus. Clear all of you? Okay, so why is apple called a false fruit? Because the thalamus develops into fruit and not the ovary, and because of which it is called a false fruit. Which part of the flower forms the fruit? The thalamus forms the fruit. Okay, the thalamus forms the fruit. Okay, this is going to be the last question that we are going to do. Okay, 15. Now see, what is meant by emasculation? When and why does the plant breeder employ this technique? Now students, under artificial hybridization, artificial hybridization, okay, we talk about two terms. One is called as emasculation, one is called as bagging. Now ma'am, what is this? Very simple students, the farmer wants to modify the flower or want to get the best yield of the flower. Yesterday also I taught you biotechnology. I told you how desired gene we want. Like I want see, there are two different hens, and one hen is thin but it's laying 20 eggs. Then there is one more hen which is hefty but it is laying only 10 eggs. Now I want to make a combination of both, where I want to get a hefty, thick, fat, hefty hen which lays 20 eggs, so that I can give the hen for KFC. At the same time I can take the egg and sell it somewhere. Okay, I'm not such rude students, I'm just giving example. Now if I want to make a hybrid, I need to make sure that there is no unnecessary pollen grains landing on my stigma. Correct or not? I want the flower to be having cross pollination, at the same time the best breed to do cross pollination. During this time, what is they going to do? Now today is Onam students, so all the Malayalis, all the Kerala people are going to buy lots of flowers. Okay, for 1,000, 2,000, 3,000, 4,000. There are people who buy for even 10,000 rupees flowers. Now the farmer knows that this is the season where I can sell a lot of flowers, and I need to make sure that the flowers are of good quality and giving me maximum yield. During this situation, they do something called as artificial hybridization, where they do emasculation. That is, out of the flower, they are going to remove the anther. Why they are going to remove the anther? Because they do not want this anther to put the pollen grains on the stigma. So they will cut this anther. Okay, they will cut this anther. They will cut the anther. And after cutting the anther, the next step that they have to follow is, see, they remove the anther because they do not want this pollen grains or the self-pollination to happen, or they do not want this pollen grains to fall on the stigma. The next thing that they're going to do is, they are going to close the flower. Okay, they are going to, let's say this is the flower. Okay, this is the flower and the anther is removed. Now we have only the stigma. Okay, now we have only the stigma. What the farmer is going to do, the farmer with the help of a butter paper is going to cover this flower and protect this flower from unwanted pollen grains. And after that, when the plant is ready, or when the stigma has got receptive, they will add the pollen grains. Okay, they will add, or they will dust the pollen grains on this flower and then again do rebagging. So this process is called bagging. Okay, this process is called emasculation. Emasculation is when we remove the male part or the male gamete, right? When we remove the male it's not just the gamete, it's the entire male gametophyte, everything. So when we remove the anther from the flower to protect the flower from unwanted pollen grains or from self-pollination to get the desired variety, after that we bag the plant. Okay, with the help of a butter paper we will bag the plant, and that plant will be again, when the farmer feels that okay, it is time for me to dust the pollen grains, it will dust the pollen grains on the stigma and again rebag it to avoid unwanted pollen grains to enter. This is what is called as emasculation, and this is what is called as your artificial hybridization, a technique that is followed by farmer to get better yields and to avoid self-pollination and to make better cross varieties of the flowers. Clear or not? Okay, they employ this technique for commercial purpose and to yield or better, to get better qualities of flower. So emasculation is clear, where we remove the anther, then we bag the plant, after that we will dust the pollen grains and then again rebag the flowers. Okay, clear emasculation. So I have selected few questions that which I found that okay, students might find it difficult to answer, or they do not know how to exactly answer. So I have answered all these questions. So I hope the session was really helpful, and also I give you a small pinch of a story of Onam as well for those students who do not know what is Onam or why people celebrate it. So I have given that also. So it was a very nice class. So happy Onam to all my students who are watching this session today, and also other students who are finding any difficulty, please let us know in the comments, and do think of subscribing to the channel, because we are trying to give you as much as content for free possible on this channel. So see you all in next class. Thank you so much for joining. Tata, bye bye.